at this thing and you have zero familiarity with physiology and biology, this might be a little bit confusing, and so you have to do some research on your own to kind of look at these. But this is called a phospholipid bilayer, which you most likely know. Okay, and it is a semi-permeable membrane. And then, if you're into this stuff, you probably already know that within the membrane, there are tons of different types of transport proteins, okay? And a major one, a very major one, that is in a lot of different cells, is the sodium-potassium pump, okay? So those of you that are unfamiliar, just imagine that this goes clear around, okay? This is on the outside of the cell, okay? So if you have sodium on the outside, potassium's on the inside of the cell. For every 3 sodium that go in, 1, 2, 3, into the cell, the sodium-potassium pump is going to pump out 2 potassium, okay? So we'll just put 2 potassium, and then it's pumping in 3 sodium, okay? So for every single exchange, it's going to be -1, okay? Now you also have another thing that's a lot like this with the same function. It's called a potassium leakage channel. Okay, and as the name kind of implies, potassium is just going to slowly leak out as it's needed, okay? It's not going to flood out because we don't want to change the resting membrane potential, but as it's needed, it will go out. And between those two things, the resting membrane potential of -70 is able to be adequately maintained. So what happens now for a charge to start, so let's say that, I want to snap my fingers, okay? But I think that hurts something to do. You're going to need a charge, okay? Remember, that would be the somatic motor division, or efferent. You're going outwards from the central nervous system to the muscle, okay? You're going to want a charge, okay? So I'm not going to go back to the beginning of this. We're just going to start here where we are to keep things on track. But what's going to happen is a charge is going to start developing inside the cell, okay? By an influx of ions, okay? Now, let's see, where do I want to do a chart here, but I want to make sure that it's very visible. I think I'm going to erase this over here. Chart. Oh, sorry, I'm just making sure this camera is still going, okay? Which it is. Did a video before and I talked for like half an hour before I realized it was off on my cardio and circulatory system anatomy, and then ended up just posting it because I didn't want to redo all that, and I'm new to this. Now what was I getting at? Okay, let me backtrack here. So the charge starts here. The axon hillock. Oh yeah, I remember what I was going to get at. I'm going to do this chart here. Okay. If we have -70 here, okay, that's the resting membrane potential that I was talking about. Now this is going to be -55, and again, we're going in millivolts here, okay? We'll talk about what that -55 is in a minute, and then we'll go plus 30 millivolts, okay? All right, so at -70, so when you start getting an influx of these positive ions, the inside charge is going to happen, okay? So you're going negative and then you're going positive. You're going to be creating what's called the depolarization effect, okay? So remember, depolarization, that's a word you want to know, depolarization, okay? So it will become more positive. Now there's different things that are happening inside the cell all the time. You know, negative things are coming in, out, positive, in, out, okay? And it's going to cause slight fluctuations in the charge, okay? Now, so that's what we call graded potential, okay? Just little random variations, okay? That doesn't ever quite reach -55, okay? -55 millivolts, we come down here, is what we call the threshold value. Okay, that's what you need to reach to make the action potential occur. Okay, so if you get enough charge to where, boom, all of a sudden it hits negative 55, what happens, remember I was talking about all these channels inside the membrane, you have sodium channels that are voltage gated. Now, what do I mean by voltage gated? Different channels are open based upon for varying reasons. With a voltage gated channel, the charge has to reach something in order for it to open. And in this case, that's negative 55 millivolts, okay? So when it reaches that, all of a sudden, the charge goes boom, and we're gonna start skyrocketing. I'm gonna put this here to keep track of where this is, and also here to keep track of the resting membrane potential. Okay, all of a sudden, it's gonna skyrocket, okay? Because all of a sudden, all right, if I take a blown up picture right here, let's bring this over here. Okay, I hope you can see this, okay? All right, I'm gonna do this, boom, 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 boom. All right, all of a sudden, all of these sodium voltage gated channels are gonna open, okay? And what's on the outside, okay, as you know, sodium. All of a sudden, this stuff is going to rush into the cell, okay? So remember, at negative 55 millivolts, these channels open. At a resting membrane potential, they're closed, so that sodium isn't rushing in there all of a sudden really quickly, okay? But as soon as it hits that, those open, and then bam, we get a massive influx. And that's what we call that the threshold value. As soon as we reach that, that is when the charge will shoot through, okay? Now, as soon as we hit positive 30 millivolts, a little bit better, the sodium gated channels are going to shut, okay? Now, different ones shut in different ways. Some of them have this weird ball and chain, like the ball kind of sticks up in there. And other ones just act on the voltage itself, so if the voltage changes to what's necessary, it will close, okay? So these are all going to lock. They're going to be closed again. No more sodium influx. But just before those close, you're also going to have, I wonder if I should use a different color here for this. Maybe you can use red. I'm hoping that this shows up good. Now, just before those sodium channels close, the potassium channels, which are also voltage gated, are going to open, okay? And that's going to allow potassium, which is already on the inside, to start floating out. I should have made that red. Sorry if that's confusing. Okay? So, because the sodium channels are closed, and then potassium starts to go outwards, your voltage is going to all of a sudden start plummeting. Okay? You're going to start going back to negative. Okay? It's actually going to go so far down, and potassium is going to lead to the point where it actually hyperpolarizes. It's going to go below the resting membrane potential to about negative 90 millivolts, okay? Almost negative 90. Okay, and then it's going to come back up until it reaches the resting membrane potential. So I talked about depolarization. Depolarization is going up. Okay, when the potassium channels open and it starts going out, you're going to have a repolarization effect. Okay, it's going back. Repolarization. Again, some important words. You probably need to know them, especially if you're taking a test on this. Now, when it goes below the resting membrane potential, that is what we call hyperpolarization. Okay? So depolarization is causing the effect to occur, the stimulus, the firing. When you have a hyperpolarization effect, it's an inhibition. It's inhibiting the firing. And the body actually uses that for different things, okay? Interesting side note, when you're looking at certain drugs like lidocaine, local anesthetic, things that inhibit pain, what that is actually doing is blocking off these sodium channels, and so sodium is never able to get inside to create that depolarization effect. And if that depolarization effect doesn't occur, then that means there's no firing. There's no firing to the brain, which means there's no pain for the brain to interpret. Pretty cool. There's also some disease related to this, like cyanide poisoning and things like that. We'll talk about at some point, I'm sure. All right, let's see. Where am I? Let me look at my notes here. Not get too far ahead of myself. Okay, if you're taking a test, a term that you'll definitely want to know is the all or none principle. That's a big one. The all or none principle is what I was talking about here, where it has to reach the negative 55 millivolts. If it's before that, it's just graded potential. It's just kind of bouncing around in there and it's not firing. But as soon as it reaches the negative 55, then bam, you have a firing. You have firing going on there. Okay, so all or none principle, definitely one you'll want to know. Okay, let's see what else. Another thing to talk about is the strength of the stimulus. Okay, normally you would think strength of the stimulus would probably be related to amplitude, but they're actually, positive 30 is all it gets to. Okay, it stays the same. So what happens is the strength of a stimulus actually is interpreted based upon the frequency. Okay, so if you have firing like this, it's firing, okay, and then all of a sudden it goes like this, okay, the amplitude is the same, but the frequency is a lot harder. Okay, so if this were related to pain, this would hurt a lot more than this. Okay, or if it was like really fast. Okay, now there's a point where they can't interconnect. Okay, so let me make a little bit more room here again. Okay, do I want to do that? I'll try making it down here in this corner. Hopefully it will show. Okay. I want to talk about absolute refractory period. I think this is probably going to be the last thing we're going to talk about, okay? Refractory period and the relative refractory, man, that's a noisy board, period. Okay. Excuse me. I'm going to try using a different marker here. It's not working very well. Gosh. So here are where the sodium channels have opened up. But the sodium channels are already open, and it's impossible to create another stimulus during that time because, well, the sodium channels are already open. You can't reopen what's already open. Okay, so I hope I'm making sense there. So within that time period, you have what's called the absolute refractory period. Okay, so about here to think about here. Okay, don't quote me on this part right here. I think it's when the hyperpolarization when the hyperpolarization begins. But it definitely starts here. Okay, so when the sodium channels are opened up, the firing is already happening. Okay, so you can't initiate another firing to happen when the channels are already opened. Okay, so your frequency can never be so fast that those overlap. You cannot make another firing rate to occur during that period. Okay, but you do have what's called a relative refraction period, and I can put this in red here. That is in this hyperpolarization area. Okay, now by relative refraction, we mean it's very difficult to actually do it, but it is possible because while most of the sodium channels, they're in the process of closing. Okay, so some will be open, and also your potassium channels. Back up there, I'm getting lost. Okay, so the sodium channels are mostly closed. Okay, but remember how I said the potassium channels were open. Okay, so if you're going to have another firing stimulus occur, you're going to have to counter the charge of that potassium. Okay, and that makes it really difficult. Okay, So you're going to have to overcome that potassium. So it is possible to all of a sudden, right here in this whole hyperpolarization effect, to have another one. But it's going to require a really strong stimulus. Okay? Let's see. Anything else we need to go over? Yes, actually, I forgot. I keep talking about these myelin sheaths, and you have no idea what they're for because I haven't talked about it. Okay, myelin sheaths, and this is the last part, help us do what's called saltatory conduction. That's another important word that you'll want to know. Saltatory conduction. Okay. So imagine up here, okay, this is the axon. So this whole thing with the sodium coming in is like it acts upon itself downstream. In other words, sodium rushes in, that's going to cause the voltage gated channels next to that area to open and the potassium channels to open later. And then the charge there is going to go on down. It's a process that acts upon another and it goes clear down the axon. But different types of axons and their diameter and also these sheaths make a difference in the speed of that conduction, okay? So different actions have different speeds. So for example, I don't know the exact speed of every single thing, but when you're moving a muscle, that's an extremely fast speed, okay? And so the axons in the peripheral nervous system for that are going to have these myelin sheaths. And they work because these sheaths cover up the axon on the inside, making these channels pointless, okay? So in other words, the charge here, let's say your charge is here, rather than going down the whole thing, opening sodium channels, influx, that charge opens more down, the charge is going to go, it's going to jump over here. All right? So it's actually skipping over all these myelin sheaths, and that speeds up the firing rate dramatically, okay? So saltatory conduction, okay? With that, I think that's a good point to kind of end it here. It's a good overview of how the system works. Next time, we're going to get into the synaptic cleft. Very exciting. That's the cool part of the nervous system. It's the most fun to actually get into, okay? With that, I think that's it. Thanks for watching, guys.